led by Ryerson University in partnership with McGill. What we do is we place graduate students from McGill and Ryerson across science and engineering in medtech industry internships, and that is towards the end of their degrees. Beforehand, we give an industry-focused course in various aspects of the medtech industry, self-paced online course, and this is for any student interested in the medtech industry. The program also includes seminars like today, as well as Job Shadow and Hackathon. So I definitely want to thank all of our distinguished panelists for being here. Brief introduction. So we're joined by Dr. Caroline Wagner, Assistant Professor of Bioengineering at McGill, Dr. David Junker, Professor and Chair of Biomedical Engineering at McGill, Dr. Don Shepard, Professor and Chair of Microbiology and Immunology, also at McGill, and finally, Mr. Mark Saab, Founder and Managing Director of BML Technology. So our panelists definitely span the range of academia and industry. I think the focus of today's panel, to align with the BBME Symposium, was going to focus on research, but I really want this to be a wide-ranging conversation for everyone to discuss how they're combating, quote unquote, COVID-19. So I think we'll start with, if everyone could give a brief intro of yourself and just a synopsis of how you are, quote unquote, combating COVID-19. So I think we'll start with Dr. Don Shepard. Thank you very much.
Thanks. So is this you want us to give our 10-minute presentation now, or do you want a 1-second introduction?
Let's start with the 10-minute presentations, I think, and we'll go one by one.
All right.
All right, thank you.
So I'm going to share my screen then. Can you all see slides?
Yes.
Presentation mode, life is good.
All good.
All right. So this was my official title, was supposed to be Pivoting to Combat the COVID-19 Pandemic, but I've given it another title. My other title, if I can advance my slides, is instead of the usual what I did during my summer vacation, this is what I did instead of my summer vacation, which has led, of course, into the Groundhog Day that we're all living through right now. And also leads to the fact that the #1 question I get is, when is this over? So what I spent most of my time doing at the beginning of the pivot was actually through my job as running MI4, the McGill Interdisciplinary Initiative in Infection and Immunity. And if you want to define performance anxiety, stand up in front of the world in 2018 and say, you're going to start an interdisciplinary group focused on infection that will be ready for the next pandemic less than a year before the next pandemic smacks you upside the head. So that was our lived reality. And of course, we had said that our goal was to work on rapid and strategic responses across the McGill community to threats like COVID-19. So I think, we didn't do a terrible job of this. We actually got rapid, no problem. So early on, you can see the dates here. We actually bet on the fact that SARS-CoV-2 was coming to Quebec, Montreal, Canada, and around. And we actually put out calls for proposals to activate our community in January, before we even were recognizing there were cases on Canadian soil. And so that was our first sort of rapid response, and we grew that into this emergency coronavirus response fund, which was supported by a very rapid philanthropic drive led by the MUHC Foundation that brought in initially $4 and eventually over $7 million in funds that were designed to keep researchers going and ramping up when the rest of the university and the world was ramping down, and to make sure that we were keeping our focus on potential solutions for COVID-19 and the public health impact of COVID-19. And you can see the community really did mobilize with over 170 projects submitted to us and almost 70 of them supported. And these were in every area that you can imagine, from modeling to therapeutics to vaccines and trials, all across the spectrum of things related to COVID-19. And the initial 3 months, I can tell you, there were 20 hours a day getting all this stuff up and running and moving. And then things have taken on a life of their own and gone off in different directions. And two of the most important things I think we did when we pivoted were we changed our existing big research platforms to focus on COVID-19. So we'd had a clinical trials platform that was looking at things like HIV studies and new drugs and therapies for multi-drug resistant bacteria. And we pivoted that entire thing at the MUHC and the JGH to doing clinical trials for COVID. And that has been incredibly successful and allowed us to offer a clinical trial to every single person who was admitted at those hospitals. And remember, at the beginning, when you had no therapies whatsoever available, the only way anyone was ever going to get a therapy for COVID was to participate in a clinical trial. And those have led to tons of papers and publications, sadly, of course, most of them saying that the drugs don't work. You shouldn't take hydroxychloroquine, for example, but there were a few wins and hits, and we've all seen the drugs and the antibodies and now the vaccines start to roll out as science has actually delivered on its promise. The other big platform we supported was the level 3 labs, first at the MUHC and then downtown. These are the biosecurity labs that let people in academia and industry test new drugs in test tube models, in animal models, and actually see if we could control the infections. And of course, you just can't do this on a benchtop. These were level 3 pathogens, and you had to dress up in spacesuits like this individual here in order to play with them. We had a TB facility that was level 3, and within 2 weeks, we pivoted to being one of the first licensed SARS-CoV-2 facilities in the country. And then we expanded that to the downtown campus, and now we are one of the leaders in biocontainment for SARS-CoV-2 in the country. And then, leaving aside rapid, we also moved into strategic. And we actually kept a small pot of money aside for areas that we saw gaps. And David Juncker, who's one of our panelists today, actually was the recipient of one of our strategic grants looking at developing novel diagnostics. The other big place we looked at and tried to get ahead of the curve was when it looked for sure like we were going to have vaccines in the early fall. We recognized that we would be exactly where we are today, asking the question, is it going to be vaccine quantities or vaccine confidence that's going to limit the vaccine rollout? And so in November, we put out a call, and by December had 6 projects running dealing with vaccine confidence and vaccine hesitancy to improve the vaccination rollout, anticipating the problems where we are today. So that was a kind of our strategic lens looking at the problem. So that's what MI4 did. We were really involved early, the catalyst, getting things moving, keeping people working, and putting our fingers in the holes in the dikes where we saw some leaks. And I think a lot of those people have been incredibly successful and now are running these large-scale independent projects, and we're super proud of their work that they are doing. Outside of MI4, I actually personally contributed in a couple of other ways and changed what I was doing from working on my own little fungal research world to this bigger COVID space. And that was through participating on this Canadian task force for COVID-19 therapeutics with a number of other McGillians that you see here, and in providing science briefing for other levels of government, both at the level of parliamentarians through Research Canada and direct science briefings at the provincial level of the opposition parties, of which I've done quite a number. So this is kind of the advising government level, but then I've actually been quite active, as many of you know, at the outreach and advocacy level within the McGill community, YouTube videos and many presentations on Q&As. Gave one this morning, for example. And then in the media, advocating for where there are things that we think don't make sense, like with this editorial that David wrote and that I edited basically on the role for diagnostics, the use of monoclonal antibodies, some of the public health measures such as the Christmas plan not making sense, roll out or opening up when we shouldn't be or closing down when we shouldn't be, and generally the overall public information and advocacy. And so that's actually chewed up a substantial amount of my time. And again, I've given 2 media interviews just today alone discussing aspects of the vaccination rollout and vaccines and variants. So the last slide here, my moving it on and handing over the microphone, is just to remind myself to mention that when I wasn't doing those things, I actually carved out a little bit of time to work on the wards and actually care for patients with COVID-19, because I am a physician. And I think this was actually critical in helping me personally understand the relevance of some of these issues. And I've shown you that Marcel Baer and Marie Hudson, the other co-directors of MI4, were also doing and continue to do the same thing. So with that, I'll pass it to whoever's next in the pipeline.
Thank you, Dr. Shepard. That was excellent. And I think you illustrate what I was trying to get across, how research is only part of the equation. Public outreach and administration are very important to combat COVID-19, and it's nice to see from our academic leaders. So let's go to the industry side of the equation. So Mark Saab, please tell us how you are combating COVID-19.
Great. First, thank you, Jesse and Sabrina, for inviting me. And hello again, Don and David. It's nice to meet you, Carolyn. I've actually seen your work, and it's nice to be sharing the panel with you. So, it's funny. I met Don, I think around 2018, Don, when you were kicking off that work with MI4, and we were at an MUHC fundraising event. And we were having a glass of wine, and they introduced Dr. Don Shepard, and he said that, the next viral pandemic is heading our way as we speak. And medicine as we know it will change forever. And I thought, wow, I thought we were going to have wine and cheese and network, and it, jokes aside, it shook me. And I still talk about that night, and I still talk about the impact that presentation had. What Don didn't tell me is that medical device at the same time would change forever and never be the same. And I guess I should have made that link, but I was probably worried about my own life for a few minutes at that time. But maybe a little bit of background on me. I didn't prepare any slides, Jesse, or anything, but I run BML Technologies, a biomedical engineering firm here in Montreal. We help companies develop medical tech. I did my grad work at McGill in biomedical engineering, actually, before there was a bioengineering. And before David was the chair of the department, so around 20 years ago. And I feel very lucky and very fortunate to have chosen this area because A, it was extremely rewarding, as I'd hoped it would be, but B, it is exploding right now in a way that we probably, maybe could have imagined, but not predicted to this extent. And the pandemic has, in every sense, taken that to another level altogether. So as you said, Don, medicine could change forever. Medical device has changed forever. And at the beginning of the pandemic, like everybody else, we just weren't sure what was going to happen and how we were going to respond to it. We were fortunate to be involved with another McGill related project with the Montreal General, the Code Life Ventilator Challenge, where hundreds and hundreds of teams and thousands of engineers from all around the world contributed designs to create a fast, inexpensive, and easy to produce medical ventilator. And I was invited to be part of the advisory committee. And it was a phenomenal experience because it was very intense and it was also highly collaborative. And it was really about mobilizing everybody's expertise and putting the right people together as quickly as possible to, get these designs to potentially seeing the light of day, which was, it was a great success, and Happily Robotics won the competition, and now they're in the second phase where they're developing the manufacturing. You can see how long these things take. That was a year ago. After that experience, I still had the feeling of wanting to contribute and wanting to kind of have that all hands on deck and collaborative approach. So some colleagues and some partners and I launched what we called the COVID-19 task force. It's a very, very common title I learned very quickly after, but David himself is part of that group, and we've invited Don to speak a number of times. And we really put some, experienced professionals in the Montreal area, but also with a Canadian footprint, with varied, varied interests, varied mandates, and varied experience to collaborate to solve problems. And I'm very happy to say that we did some, very exciting work. We worked with NGen early on. We ended up helping an NGen project called RRP Canada, which is a nationwide PPE connector of suppliers and users. So we helped them get the regulatory matrix that they needed in place to understand what kind of certifications these companies would need. And then we worked with the NRC IRAP collaboration with ICED Canada to, again, help with the regulatory aspects of developing test swabs on the millions per week scale, which was a phenomenal, phenomenal endeavor. And we're happy to say that that's rolling now. We didn't have a primary role in that, but we were able to help with actually a parallel here with what we do every day, which is, like I said, briefly, to help companies develop medical tech, but more specifically, that's intervening and helping with things that are quite convoluted and quite complex in terms of collaboration between technology development, clinical application development, and clinical validation, and all the regulatory affairs that are inextricably linked to that, and absolutely mandatory for bringing these products to market. And all of that kind of varied experience and varied existing workflow was really relevant to projects that needed to get developed quickly to help with the pandemic. And then since then, the task force has been focused on rapid testing, and maybe David will talk more about that, but we're interested in educating and supporting the projects that are looking for advocacy and understanding and demystifying some of the confusions. Like I said, I'll let David comment. I'm sure he's going to talk about rapid testing. But in terms of, as I said, medical device and what it looks like today, we'll get into it during the panel, but it's significantly more exciting and significantly more intense than it was a year ago. I could say that. So thank you.
Okay, thank you very much. Let's move on to Professor Wagner. Pretty new professor at McGill in bioengineering, and when you came, you had to pivot your research right away, it seems. So please tell us, how are you combating COVID-19?
Thanks a lot for having me. I'm really happy to be here, and I feel a little ridiculous being on this panel because I think that these people have a lot more experience and influential roles than I do in all of this, but I'm happy to talk about what I've been working on. So yeah, the pandemic hit in the month after I came back from maternity leave with my first kid while I was a postdoc, modeling all sorts of, at the time we cared about more diseases than COVID-19, so we were modeling all sorts of different things. And so everyone in my group, I was a postdoc at the time, switched gears pretty fast to thinking about, what can we do? And I think a lot of research groups around the world felt similarly, even people who don't study epidemiology or disease dynamics. I think people from engineering fields, people from all backgrounds started to say, like, what does what I know, how can that help in some way or another? So I'll talk. I prepared some slides, but I feel like they're a little bit like detailed, but I'll zoom through them quick and just give you a bit of an overview of what we've been thinking about and why I think, and sort of what we, in the context of this problem. So basically we said, okay, look, we know there's these classic disease models that exist, and I don't want to get into the details, but this is what's called an SIR model, where basically the world is split up into susceptible people, infected people, or recovered people to a disease. And so you can do the math and see how all these things work out. And this is where this term, this reproduction number, R naught, that you've heard all about at this point, comes up. And this model says, okay, if your reproduction number is sufficiently small, you're not going to have an epidemic. If it's large enough, greater than this value 1, you get the sort of predicted epidemic cycle. Although not everyone gets infected in these classic disease models, eventually enough people are immune that things start to die out. So we said, okay, we know sort of that aspect, that's sort of well established. And then the other side of this coin that we were thinking about really in the early stages, so we're talking like March 2020, April 2020, was, well, is a vaccine going to come eventually? And at the time we thought that 18 months was like optimistic in terms of vaccine development. And so we were really blown out of the water when, 10 months, 8 months down the road, there was already things in successful clinical trials looking like they're going to hit the market pretty fast. So we said, okay, let's think about also, what would a vaccination do? So what happens if you can now move people from the susceptible to the recovered class without having to have them get the infection, which is obviously the problematic part of this journey, let's say, from susceptible to recovered. So again, without going through the details, classical theory tells us there's some kind of a curve that exists between the actual fraction of the population that you'd have to vaccinate to eradicate a disease, so to have no cases at equilibrium, versus how transmissible the infection is, or how large its R naught is. So it turns out something like flu is expected to have an R naught around 2, so that would say you have to immunize about 50% of people to not have cases at equilibrium. But then something like smallpox, which is actually the only human disease that we've ever managed to eradicate, has a higher R naught of around 5. And so there you really need to get a lot of people immunized in order to get rid of infections at equilibrium. Measles, for instance, has an even higher R naught, so it means you really have to vaccinate a lot of people to not see transmission, which is why hesitancy is a big problem also in terms of measles outbreaks that we've been seeing, especially in the last 20 years or so. So the question was, where's SARS-CoV-2 on this curve, and what does that mean about how we have to start thinking about what vaccine rollout will look like? People have estimated it to be around 2.5. which gives you like this classical result from this very simple equation, 1 minus 1 over R naught of about 60% that I think a lot of people have jumped onto. But in reality, this is going to be really sensitive to a lot of things. I think back in March 2020, variants weren't on anyone's horizon. We didn't know anything about this infection, and now everyone's talking about variants, right? So these numbers will depend on how the virus changes over time. How will that affect its transmissibility? It'll depend on the fact that we're all not just little gas bubbles running around bumping into each other, homogeneously mixing, but we have spatial structure where we're segregated into populations. People are different inherently. So there's a lot of things that will really challenge what these numbers mean. And at the time there were a lot of unknowns. There's still a lot of unknowns, but I would say there were probably even more back when we started thinking about this. So what we said is, okay, we know what these classical models tell us, but we have a lot of things that we don't know in terms of what to plug into these models. So let's think about the fact that at the time we said that probably the one thing that we were grappling with was, well, an SIR model says that once you get the infection, you're immune for life, but we don't know if that's going to be the case for SARS-CoV-2. So our first question that we asked was, what happens if you aren't protected forever? So there's all sorts of, you can go into details of how you are. I'm not going to talk about this too much, but we said, okay, we could sort of, maybe what we could do is turn to other related viruses and say, what does protection look like from those viruses? So there's other beta coronaviruses, including common cold viruses that people seem to get infected with repeatedly. And we sort of started to constrain ourselves in a parameter space and said, well, let's assume protection is lasting on the course of many months to maybe a couple years. What does this tell us about what might happen down the road? Because what these models predict is that there's a big difference, and I'll show you just schematically what this looks like here. A model where people are immune for life looks very different in terms of what the predicted outbreaks look like over time versus a model where people continuously can be reinfected, which is something like SIR, but now S. So you are recovered, but now you're moving back to the S category. And that's because in an SIR model, you have to wait for people to be born into the system for there to be enough susceptibles, and then you can get an outbreak. But in an SIRS model, if you're constantly replenishing the supply of people, you can see a lot more periodic and repeated outbreaks. So we just asked ourselves at the time, this was our first question, what does it matter if this infection is fully immunizing versus partially immunizing? And that was sort of the first road that we went down. And so we presented it as like scenarios for the future. I think we called them futures, I think was the word we used, where we said, let's imagine different scenarios. Let's imagine a really great world where people have really robust immune responses and really almost never get reinfected. How does that change how we think about what epidemiology looks like versus a world where maybe reinfection happens very often and you aren't very protected? And then we said, now let's add vaccination to this picture. What if we have a really great vaccine versus what if the vaccines aren't very protective? So we started to sort of put out these guidelines. I'll stop sharing for now, or not guidelines, but sort of guesses for what we could imagine based on the fact that we didn't have a lot of information at the time. Can't stop sharing for some reason. So this work anyways translated, I don't know what's going on here. I can't stop sharing. Okay.
At the top, maybe?
Yeah, it's because I have you guys in.
Don't worry,
Thank you. Thank you. So that was sort of the first question. And so like I said, this was sort of fall 2020. And then all of a sudden the question wasn't, well, what if we have a vaccine anymore? The question was, now we do have a vaccine, but should it be given as 1 dose or should it be given as 2 doses? Should we space out those doses? How much should we space out those doses? How good is 1 dose relative to 2 doses? So what we did then was sort of extend this to think about the implications of multiple dosing regimes. And what we're doing now is we're saying, okay, we sort of have a better idea on how good these vaccines are. We're starting to get a better idea on how long immunity is lasting. now, and this is a question that's occupying us and a lot of people, I think, these days is, and there was actually an XKCD comment about this, which I kind of frustrated me because I usually really like XKCD comics, but basically it was a calendar and it said, you know, March 2020, and then every month was the same, and then people got vaccinated, and then it said May 2021, like, yay, everything's back to normal. And I thought this was a very problematic picture, because maybe that's the reality if you happen to live in somewhere like the United States and you had great access to an effective vaccine right away, but that's not going to be the reality for the vast majority of people around the world. And I think that what has to be recognized is that, you know, if infections are allowed to persist in other places, then inevitably that's going to mean that the potential for new variants to pop up that potentially could come back home and the vaccines could not be as effective too in the first place is a reality that everyone's going to have to grapple with. And so how we think about sharing vaccines between different populations is a huge question, and one that we have to answer as soon as possible. So I think you can see that what I've been doing was sort of like just me in my room, like coding, and it's a little bit more theoretical, and in some way it's not grounded in data to the extent that I think some of the other panelists or what they've been modeling has been related to. But I think it has led to sort of giving some directions for how to think about these very open-ended questions. And in terms of the, what the end result has been, I have, like, on my end, that's looked like talking to panelists with people who know a lot more than I do, or like groups from the WHO or groups of other modelers, and sort of explaining what we've done and giving them some context for that work. And then ultimately coming down to those people making the decisions that they will, either as these international groups or in different countries. And talking to media about this stuff, which I find extremely stressful. I don't know how Dawn does it regularly. Every single interview, I have a mini panic attack beforehand. So stuff like that. And I think that's been a dimension to this job that I could never have imagined before the pandemic hit, that's for sure. So I'm gonna stop talking and I'll be happy to talk more in the questions.
Excellent, thanks so much. Well, you're doing great in front of this live audience, so thank you. All right, so last but not least, Dr. David Junker, Professor and Chair of my home department, Biomedical Engineering. Please, how are you combating COVID-19?
Yeah, thanks. Thanks, Jesse. Thanks, everyone, for participating. I mean, I didn't really have slides prepared, but I saw Dawn making such a nice slideshow, so I want to bring something up too, which is why my cover slide doesn't really match the topic, but I was also preparing another presentation. So and actually, well, I think there's two aspects. One is the research in the lab, and as Don, our funder, has want to giving back some data here. And then there's also the public use of rapid tests, which is really tight. I mean, on the public outreach that I have ended up doing, because I think there was an important message to be given, and because of the potential and the need for rapid tests, as we just heard from Caroline, it's probably testing is something that's going to stay with us for a long time as well, because not everyone is getting vaccinated and variants are coming and so forth. And so I'll try to quickly go through this. So, and it's going to take you a little bit here, because our lab has a lot of microfluidics, or so-called lab on a chip. And however, a lot of these lab on a chips, they're more lab around the chips. They need peripherals, a computer for like making automated systems. And so what our lab has been working on a lot is capillary microfluidics. But there the question really is, how can we control the flow? And versatility, how can we scale it up, and how can we make it reliable? And to some degree, this is already a very successful technology, and actually, particularly in the context of COVID-19, among the first personalized test, and also maybe not the cheapest personalized tests, and the one that are fastest are rapid antigen tests. And so they're the ones that have this big potential, and that some places recognize it and some places are having a difficulty to recognize it. I'll come back, I circle back on that towards the end of my presentation. But so what we have been working in the lab is to try this capillary flow more versatile. And the way we have started to think about it, the way we have started to think about bringing this energy and the control into our thing is through chain reactions, which probably through nuclear chain reactions, which we also know through chemical chain reaction, which are microscopic, and which can a way of controlling the events. But the way maybe that was more inspiring, more close to what we're doing, is something more like this kind of chain reaction here, where basically it's one event is triggered by the event preceding it, which then triggers the event that follows it. And so here we have potential energy that is stored and that is kind of delivered in a controllable manner. And this is what we're trying to implement in a microfluidic scale, where we have a series of reagents and the delivery of one reagent is conditioned on the emptying of the previous reagents and so forth. And this is kind of videos, we've made some really nice videos to illustrate that. And the way we've implemented that, we have liquid reservoirs here, and then they have these air connections in between, and there's an air from the outside. And as the flow goes, only the first one is connected to the air, and only once, and so capillary forces play a big role here, only once this is connected does it connect to the next one here. And only when this one is opened up does it connect to the next one. So we have this chain reaction that can then propagate throughout this chip, so that we can now control many events just by having individual or localized control. And so this is first demo, we can scale this up. I think that's a demonstration now of the larger scale chain reaction, where, and that's really credit here to students from my lab, Azin and Ahmad, who worked really hard for doing that. So here we even chain the chips together, so we can have chain reactions across multiple chips, but really to demonstrate here the scalability of this process. And here it's how it's going from one row to another one, where again, we have this air link opening here, and then that can propagate from one chip to another. And yeah, we speed it up only quite a bit here. So this is like 5 times. So you can get a sense of that this can be applied to a larger scale. And so this is a technology we think is very powerful, because now you can, what normally you do the computer to do, you can now encode into this, you can encode the function of the liquid algorithm into the structure of a chip. So instead of encoding into a computer, you encode it now into this microarchitecture. And this is the technology that we now wanted to use for COVID-19. And here I have another nice video where now we have a chip. And so this is a chip where now we do similar to this lateral flow, to the flow I should show you before, but here we really replicate what's a classical ELISA test. We have all this incubation washing onto the chip. And so we can have then the reagents and a better control and better washing and hopefully better sensitivity than this simple lateral flow assay. And so here it works by basically connecting the chips together. So this is an antibody test. Here we're testing whether a person has antibodies against COVID-19. So this would be the sample, the yellow one. Here's the buffer. Then we'll have a detection antibody. And so you'll see it here in the graphic illustrated, which binds now this antibody. So if you're positive, it will bind here. If it bounds here, then we'll bind streptavidin. And then we have another rinsing step, and then we'll have an enzymatic amplification step. And as you can see here, it actually shows the enzymatic amplification. So it gives you a tool type of line, like on these classical lateral flow assays, but with all the added washing step and an enzymatic amplification step, which you can't really implement if you have everything pre-dried. And this also has the benefit in the way we implement it, that this is actually a permanent record, so you're not time constrained where you read it out. And so, of course, this is still more work to do. So we want to pre-dry our reagents, for example, so that the user would have very little to do. But what we're showing here is that we can have a very nice readout, a quantitative readout, and this is actually done during smartphones, so that we could potentially have quantitative results at the point of care. So this was an antibody test, and that was actually the focus initially in the disease. And people very wanted to know whether people would have immunity, so that we could evaluate how many people would have been infected. And this is, and so as the pandemic started, we really pivoted that, and this is all the people then who changed and worked on this project. And then the second project, once the interest in actually this immunity waned, we shifted towards antigen tests, which is the one I showed you. And the antigen test, as we started working on that, and that's where now suddenly the public outreach aspect came in, where I came out of my little ivory tower in the duff, if you may call it that way, to speak, is that we start reading, and we have this paper here by Larimore and Mina, who basically said that, you know what, you don't need to make a very sensitive test. I mean, because that was one of our challenges. We need to make a really sensitive test to be able to diagnose these people. But they said that, no, you don't need a sensitive test. You actually need a test that's frequently and and have a turnaround time that's very fast. And that will actually be much more efficient in turning down the spread of the pandemic. And this can be explained basically by the time course of the viral infection. So when you get an infection, initially, of course, you have an incubation period where you have very little virus and you're not spreading it. And one of the reasons why this virus has been so effective is that actually start transmitting before you have symptoms. And this is really this is actually one of the big surprises or bad surprises of this virus. But, and where we're a bit safe, is that actually the virus is not that infectious. It takes 10 to the 6 viral particle around that to actually to infect someone else. That's the threshold that people have devised through lab experiments. And so if you look at this curve, is the period where you can infect is this blue curve, which is actually very nicely aligned with the detection limit of rapid antigen tests. And also what's interesting to consider, this is a logarithmic scale, like 99% of your virus is during these few days. This is where you're most infectious. And so what this shows that rapid antigen tests are actually, and that's a bit of coincidence, the sensitivity is really well aligned and almost perfectly aligned with the ability, the having live virus and so being infectious. And so what people often compare these rapid tests is, well, we have PCR tests, they're less sensitive as PCR tests. And this is exactly where now we have to distinguish between a medical test where you want to know if someone has been infected and the public health test where you want to know if someone is infectious. Because when you do PCR tests, they will detect residual RNA in the sample days after you actually have had the disease, at a time when your likelihood of infection someone is very, very low. So really, if you want to know if someone is infectious, this should be a negative test. And on the other hand, the antigen test will actually be much more tailored to. And so if we recognize now, so if you want to use a test to limit the spread, then having a test that actually tells you if you're infectious and also having a fast turnaround time is really what will make the difference. And that's why we should be rolling out these tests very quickly. And so one country that has, for example, done that was, and actually I didn't bring in the graph here, was Slovakia, where they did one large massive testing on their population. And when they did that, the viral curve was growing, it really abated basically for a week or two while they did the test, while it grew again. And now they actually did a second round of rapid testing where, again, they have been, so also their vaccination rate is lower than ours. They have had a suppression of the virus that is actually much higher than ours. And they said that ours, so they have a population of 5 million. And I was just communicating with the public, the chief epidemiologist, and they had 250,000 tests that were 250,000 cases discovered by rapid tests, and about more than half of them on asymptomatic people. So in Slovakia, everyone knows someone who has basically had a positive rapid test result as a way of suppressing the spread of the disease. And so basically that's the story of, I had these early results, and so in the US, Michael Mina was actually having a strong outreach via Twitter, which is also another tool I discovered as part of this episode, on how we should deploy these rapid tests. And no one was talking really much about this in Canada. And so I started to spread that message. And via, like Don just said, we are trying to write op-eds, via also interviews with journalists, via live interviews as well, and trying to get this to move the needle. And I think we had some successes. So at the federal level, they developed now, whereas they had a task force for immunity, therapeutics, which Don is on, and what is it? I think the vaccines. They didn't have anything for testing. So testing really has been a bit the neglected child here. So the federal level then developed a panel for rapid testing, which then made also recommendations in line with frequent testing. So that has been a big progress. We have seen now some provinces in Nova Scotia, they actually had these pop-up tests administered by non-medical professionals, which was globally world leading. And they have been one of the provinces that has best managed the COVID-19 disease. But in other provinces, the uptake of rapid tests has been very, very slow. We see a little bit movement now in Ontario, where they have these pop-up buses, but here in Quebec, for example, still completely behind. And well, so I think this has also illuminated a number of things. So one of the things is, well, science experts, sometimes they're, it's difficult to, when these new diseases come, sometimes you're taking short. I remember in the beginning, the experts were advising against masks. The latest thing again through Twitter was, I just saw today that there was a paper that showed putting up the plexiglass panels in schools was actually counterproductive, because what happens is that the little droplets, they go down and they get trapped inside of these little things, and then people pick them up more than if you had an open ventilated space. And so we see also the importance of bringing in not just medical doctors, sorry, Don, but also bringing engineers and well, in this case, aerosol scientists to really speak of the disease and think about disease on first principles basis, which I think speaks well to us as biomedical engineers. What is also exemplified for me is the dysfunction at many levels of the healthcare system in terms of responding to a pandemic like this, which is complicated in Canada because there's federal decision-making, provincial decision-making, the ineptitude, I would say, at some levels with rapid testing, and I would highlight Quebec as one of them here, because I've engaged actually with, or we have engaged with them, other people, and we talked to Aruda and others, but they, even with the example of Slovakia, they're still very reluctant to roll them out. And also one of the challenges I think that's still underestimated is actually Health Canada. Health Canada has very stringent guidelines for tests. And so I talked to rapid test makers, and they said historically they don't even try to, so they're Canadian companies, they don't even try to apply to Canada because the rules are so stringent that they can't get them. And so in the case of this pandemic, we have exactly the same issue that and I think with vaccines, we have seen we don't have local vaccine manufacturers, but we have the local rapid test manufacturers, they could not get health approval for their test, in part because Health Canada actually adopted the FDA guidelines, and as long as Trump was there, they were not very well designed as well. And they don't recognize public health versus medical disease. And now they have updated them, but still they do this comparison to RT-PCR. For example, they ask rapid tests to detect people with very low viral load, which by definition is not compatible with rapid tests. And so here it's really an important case for, I think, advocacy and also informing the public, but also keeping maybe, and it's a tight balance, because on one hand, being a scientist where you question the answers, and on the other hand, being the expert who provides the answers. But as we see science in a pandemic, it's very, if we describe science has the answer, it becomes I think, challenging with formulae. Science is not a religion, things can change. And science, to me, ultimately is really a method to describing different data, but then also keeping somehow it's both being forceful in making the case, but also keeping the humility on one end that, we don't know everything and that we have to be willing to update our knowledge as this new data comes through. And I think that's a challenging step for politicians and maybe some of the more prominent people, because once you go out in one direction, I think Quebec for rapid tests, they have consistency against them, switching back can sometimes be difficult. Also, to the credit of the current government, following Don's hammering, they often did actually reverse their position in terms of lockdowns and so on. And so, I'd say that's actually a positive outcome, that we can have an impact if we speak loud and forcefully and eloquently enough. And so I think that's a great lesson also, that for everyone that sometimes you can try to have a bigger impact, and sometimes you're successful, and sometimes, it's a bit harder as with the rapid test.
Excellent. Thank you so much, Dr. Junker. Okay, so now I have some questions on my end. I'm not sure how much time allows, but I want to go back to Dr. Shepherd. First, I want to share the comment you made in the chat here. Quote, "I want to go on record that I've refused to comment on ventilation since the beginning and told them to talk to an engineer," followed by 4 exclamation marks. So an enthusiastic rejection of commenting on ventilators. So well noted. So my question for you is, what are the most exciting or high impact areas of COVID-related research that you're seeing? Not necessarily in your research field, but across microbiology, immunology, anything in MI4 or beyond. So please.
Yeah, I think actually that what Carolyn was talking about is the real next frontier. The real question of understanding the duration of long-term immunity and how much there is, and the activity of our various vaccine-related immune responses against the variants. The data is very clear when you dive into it, that that natural immunity is ineffective against these new E484K mutants, which include the South African, Brazilian, and Indian. The studies from Manaus are beautiful epidemiologic studies. They're clear. You can go from 75% of your population infected with wild type virus to 75% infected with the new virus in wave 2. That's pretty damning evidence for failure of immunity. So I have no doubt that the Barrington Declaration people can take off their tinfoil hats and that natural immunity isn't going to save us against these new variants. But we don't have these data at all when it comes to what the efficacy is of the mRNA vaccines. We have lots of surrogate assays with test tubes looking at antibody neutralization and antibody titers, but we have no data on whether T-cell immunity is important, how long these things last, how quickly they recover. We don't have natural infection epidemiologic data to backstop these immunologic surrogate assays. And as a result, the modeling that Carolyn's doing could be incredibly more informative if someone could give her the type of real numbers she needs to say, okay, no, look, at 6 months, the vaccines are this effective or that effective against these new escape variants. And then the new wrinkle, which I don't know how many of you caught, that at about 11 o'clock last night, Moderna released the top line results of their new variant booster. So Pfizer, AstraZeneca, and Moderna have been working on variant boosters. And the results from that, at least the top line results that I got last night, tell us two really important facts. Fact one is that at 6 months, most people who had high levels of neutralizing antibody to wild type virus still had it. So this 3-month decline after vaccination, we're not seeing the decline yet. At 6 months, there's no meaningful decline of antibody titers after vaccination. But more importantly, at that 6-month point, about 50% of people had low neutralizing titers against the variants, and all of them responded to this new variant booster. So although we still don't know the real protection levels, it's quite clear that at least with the surrogate markers, these new variants that are coming down the pipe as vaccines are going to be effective. Now we got to figure out how to use them. Do we replace the second dose booster with the variant booster? Do we give a mix of 50-50? Do we give a third booster? What's the timing? Because most of us believe the 21-day timing is complete rubbish and will actually work less well than the Quebec version of 3 to 4 months. These are the questions that I think have to be answered so that we can actually look at the real question everyone wants to know, which is, when will herd immunity kick in? Because until we know what immunity is, we can't actually come up with any meaningful number because we're missing half the data set that Carolyn so elegantly pointed out. We need to do the calculations.
Excellent, thank you. And I think the idea is that maybe this will inform, our knowledge of mRNA vaccines in general for the future. So very important work. Moving again to the industry side. So Mark, what are the most exciting and high impact areas of innovation in the medtech industry that you're seeing today?
Well, the nice thing about device is that, and I'm really coming with a different perspective here, but it really gels nicely with what Carolyn and Dawn and David are talking about. And if, I agree with everybody on the panel that this new reality is not going away anytime soon. So even the need for PPE was not mentioned. But I can imagine we'll be wearing masks, not just in international context, but for years to come. So, in terms of the way that, device can support this kind of development. So, immunology and microbiology and population disease dynamics is so, they're topics that are so prevalent right now in context of what's going on. But as, another little subtlety in something that Dawn mentioned about Carolyn's work is getting the right data to the right place at the right time. That takes a lot of new kinds of technology that don't exist today. So, communication, health communication is going to change. Interoperability is going to change. You have to think of kind of the secondary layers of that if you're interested in medical device, biomedical engineering, and not thinking only of the immunology and biology here is going to take great technological advances because, and these are not anecdotes, and they are stories that the engineers like to tell, but this is a real one, from April of last year, where, maybe in the news or maybe in these conversations we hear about initiatives that are in place to help with the, transmission management, and population management, and we talk about contact tracing and things like that. But this is April 2020 when the pandemic is hitting, and there's a lot of panic, and we are literally seeing fax machines in provincial centers with paper, explosions of paper on the machine and on the floor and on the table next to the machine. And I was there with a software company, and I said, what's that? And the answer was, those are test results. And I thought, those are not just health records, those are real-time test results. And we're talking about contact tracing. We might have contact tracing information 2 months later with this kind of technology. So, it's a lovely anecdote to come up with, but it actually is a real one. And I was working with a software company that quickly implemented a platform to get the provincial government to work with contact tracing. That is an exponential change in terms of the ability to support this very critical work that's going on. So as biomedical engineers, that's the holy grail of what we're after, right? To take technology, but to apply it to a real health context and a real, kind of, how can we help our fellow man? That was why I got into biomedical engineering. I'm sure that a lot of you share that. We want to know, how can we make real impact? And there is no shortage right now of areas to contribute to. So in my career and in my lifetime, I've never seen the field more ready for innovation. And I've never seen the dynamics that are so prevalent in everything we do, not just consumer adoption, like in the consumer product world, where the new iPhone comes out every year, and that's not how health tech is developed. We talk about timelines of 5 to 8 years, but adoption is very complex in health technology. So, medical adoption, governmental adoption, population adoption, that barrier has changed dramatically. So all professionals in the field are ready to accept new technologies much more than they were, before the pandemic hit, because as it was said earlier, it significantly, massively exposed weaknesses across the entire healthcare system. So all of a sudden, it's an all hands on deck. Everybody grab a shovel. There's no shortage of dirt to go around. So
little disconnection.
I think we had can you guys hear me still?
Okay, you're back.
Okay, all right. So yeah, sorry about that. So when you look at the kind of dominant variables in our world, adoption, as I was talking about, is one of them. Regulatory barriers are things that are misunderstood until you really get to the forefront of trying to commercialize medical technology. And then you say, okay, we have to get this through Health Canada, we have to get this through the FDA. Well, those regulatory barriers have changed, not because the agencies are willing to take more chances, but because we've all been forced to kind of revisit the risk reward and risk benefit curves, and realizing that there are ways to do things maybe a little bit in more innovative ways and a little bit faster. And it's not about cutting corners. It's about focusing on what the key areas are and understanding what the real risk reward, dynamics are in releasing medical technology. And again, that has never been more prime for innovation and development. So take your pick: telemedicine, new sensor technology for remote patient monitoring, interoperability of EHRs. So again, all of this data that we're collecting, to get back to Carolyn and to get back to Don and David, well, if it doesn't come in properly, it doesn't come in on time, and it doesn't come in reliably, or there are cybersecurity risks and data privacy risks and government barriers, it's not going to work. So again, like I said, there's never been a more amazing, inspiring, and kind of open landscape in terms of medical device innovation, and it's an amazing time to be in the field, really.
Excellent. Thank you so much. So Dr. Wagner, we have a lot of grad students in the audience today. A lot of them have research projects already. But my question to you is, for those who might be pivoting the research or moving on to higher education and new projects, what advice do you have for graduate students who are inspired to do research that aims to combat COVID-19?
Oh, that's a great question. I have a history of switching gears pretty dramatically in my academic trajectory, even from my PhD in engineering to my postdoc in epidemiology. It was pretty big. jump. I think one thing that at least I've experienced from working in these two worlds is there's a dramatic, I'm going to use the word dramatic, lack of communication between different fields in science. So I think that people in the science world, or let's say the engineering world, don't read the literature from other fields, and that's true for people in the disease modeling world that I've seen. We had right now, and I think this is where for instance, the fluid mechanics stuff that I did in my PhD comes into play. I think aerosol transmission is a huge question, and it's going to be a huge question when the next viral pandemic hits too. And so understanding a bit about, like actual fluid mechanic properties of you emit droplets and they evaporate and the virus does something and then it gets into the next person. Those are like real engineering questions at the end of the day, and they're related to the whole ventilation question that was already discussed. And I think I kind of chuckled because sometimes I'd be in these rooms with mathematicians and disease modelers, and it was almost like you were reinventing the wheel in terms of describing like very well established thermodynamics and mass transfer equations that engineers are all very familiar with. So I think my advice on the one hand would be read outside of your field and learn what other people are doing, learn what approaches they're taking, because I think that that's going to put you with a huge leg up, and you'll have really good insight into what's going on. And then also what it'll do is it'll help you to understand sort of the context. And the other thing I would say is that everyone has these great insights from their own fields that stem from their own respective knowledge sets. So there is something that you can contribute no matter what your background is, because you bring something unique to the table no matter what your background is. But recognize that other people are in the same boat, right? So recognize that their background is also helping them to say something important. And so getting a grasp on where sort of this multidisciplinary aspect comes in and where you fit in, and sort of, it can help at least shape the problems that I think you could help answer moving forward. But I think the encouraging news anyways is I think there's tons of room for lots of people in this field.
Perfect, thank you. So we have a couple more minutes left for Dr. Junker, our last question here. My question for you is, what are going to be the long-term consequences of this explosion in research and attention that's being paid to COVID-19 today?
Well, that's not a loaded question. I think I'll maybe segue what Mark said. I think what we're seeing, it has had huge impacts in terms of how telemedicine has, something was considered impossible and legal barriers and so on. And well, suddenly it's here, and and it kind of works, and people have adopted it. So this is a huge change. And I think it might segue also with our keynote speaker, right? Where Mike Snyder, who developed this smartwatch, so for the ones who are not here, who basically they showed that they could pick up, COVID-19 and any kind of disease or infectious disease based on heart rate changes, and for COVID-19 up to 7-8 days before people became symptomatic. And so the integration of these mobile sensors with telemedicine and healthcare, suddenly now we can see that this COVID-19 crisis has propelled us forward in terms of what these things are doing in ways that we didn't. We took like giant steps and leaps and bounds for things that before were just like getting tangled into all these administrative and legal and healthcare requirements. And so I think that's going to be a big change. I think the research landscape, we'll see, of course, there will be an effort now here in Canada. There's also a big effort now on biomanufacturing, recognized on the lack of vaccines. That is a big change. So they're focusing now on making vaccines, but this could maybe be extended also to cell manufacturing and cell therapies as well, which I think is becoming a big part and which I think is obviously very close to biomedical engineering as well, and which has a big potential in terms of moving to the future. I don't know, maybe some of my colleagues here have something to ask. I think Don is kind of burning to provide some input here, so maybe I'll give him the...
And we can keep the conversation going, but we are at the end of our hour here. I do want to thank you. And let me say, if you want to learn more in the audience about the MedTech Talent Accelerator, please visit our website, ryerson.ca/medtech talent or mcgill.ca/BME/career. We do have an info session upcoming on May 19th for the fall 2021 internship cohort. So visit our website for more info or follow us on LinkedIn and Twitter. And with that, I would like to thank our panelists on behalf of the BBME Student Society, the MedTech Talent Accelerator, and everyone in attendance today. Please join me for a virtual round of applause.
Okay.
Thank you, Jesse.
Thanks,
Very nice.
Jesse. Thanks, everyone.
Thanks, everyone.
Thanks,
So
everyone.
if any of our panelists can stay, we would open it up for Q&A. Otherwise, no problem at all.
Sorry, I have another meeting I'm going to pop off that I'm now 1 minute late for. Take care, everyone. Stay safe.
I'll answer on your behalf, Don. I'm happy to stay a few minutes,
Okay.
but I want to give the floor maybe to Mark or Caroline as well.
I can
if
stay.
they can.
Yeah, I can stay for 15 more minutes if that's okay.
Perfect. And in the meantime, if anyone in the audience has questions, please ask through the chat. And okay, we do have a question right away from Olivia. Olivia, you could turn on your microphone. Thanks.
Hello. First of all, thank you for being there. That was really interesting and really, well, really on point. And I have a question. Maybe it's a bit more relevant to Professor Wagner, but probably Professor Juncker might also have some have some interesting comments on that. Especially for disease modeling, like I feel it's a field that didn't get any recognition before kind of COVID hit, pretty much. Like from one day to the next, everybody was talking about models and making like policies based on models. And I was wondering if there was ever any frustration or feeling that people didn't really understand the limitations of models and we're just taking them completely for granted and like for ground truth when it's only like science with its limitation and based on the availability of data and everything. If you ever felt this way when trying to explain models and things like that.
Yes, I have felt that way. For instance, when we put out the dose spacing paper, you know, in the paper you can caveat it to the moon with all the things that the paper depends on and all the assumptions you've made and recognize all the unknowns, and then the journalist who's calling you just says, is Quebec screwed with a 4-month spacing strategy? And they want a yes or no answer, right? So all these subtleties that you spent weeks carefully describing are sort of out the window. So I would say yes, that's an issue, but I think I've also been pleasantly surprised by people, you know, are receptive to learning, and there's been some fantastic science journalism, like a lot of the stuff coming out of the Atlantic and the New York Times has just blown me away with the depth of understanding and the like curiosity to actually get the right people to answer these questions. So I would say it's a mix. And then the other thing I would say is, you know, maybe disease modeling has seemed a little bit esoteric, but you know, this is an epidemic that is affecting a lot of people, but we're still in, you know, the AIDS epidemic and the HIV epidemic. There's lots of other ongoing epidemics, and, you know, disease modeling has been, you know, epidemiologists have been working, the Ebola outbreak, you know, people have been working on these problems for a long time. It's been, and now I guess it's affecting so many people so quickly that it's on a lot of people's radar, but yeah.
So yeah, I'll maybe just
I'll give Dr. Juncker a chance to respond.
So since I was also addressed, actually, no, I think I mean, this is the first pandemic in the Western world since 1918, and so I think our interest in pandemics and maybe also our legal and regulatory framework were really not tuned to that. And more specifically, I mean, this is public health, right? Public health for us is really a background question. It's certainly not for low and middle income countries. So the interest in these public health models and so on, well, they're actually the ones who have often the biggest impact in terms of public health, but they're not the ones that are in the intensive care unit typically and taking care of people. And so here, well, we have this short-term reversal, of course, with COVID-19 and this focus, because now it affects us. And I think, well, I think this is how it is. I'm afraid that once the COVID goes away, maybe this will kind of drift back into the background, and we'll talk more about AI again and whatever else the next technology innovation will be. And the question is how. Well, but in the end, what's your motivations to do? Is your motivation to be and there with the journalists and be interviewed? If that's the case, then well, I mean that's good too. But then you have to consider that. If you want to really have something that has an impact, you can have an impact, of course, too, by making these models, having them be actually used, right? And this can happen, of course, even if you're not necessarily on the spotlight.
Not to take the attention away. I know there's the hands up, and I want to get to those, but just to add to something here that really it hangs above all of this, and Carolyn, you touched on it, is what we're working on and what you're working on in your lab and what we understand in an academic or a research context is very different than what gets, than what is needed to commercialize, and very different than what is actually understood by the general public, and more importantly, sometimes by decision makers out there that are responsible for getting these phenomenal solutions to actually be used in a real healthcare context. So, Carolyn, when you feel that frustration of, somebody wanting a very simple, very quick answer, well, it's a fundamental aspect of everything we do, because almost everything we work on is very difficult for the general public to understand, and very difficult for people that are not directly in the field to understand. So a major part of what we do moving forward is always going to be about communication and about always targeting adoption at the end of the day. So just a little bit of the industry commercialization perspective, it's not what we think. We sometimes we think we're doing something phenomenal, and everybody will, it'll cause a tidal wave of appreciation, and then it's not the case, because it's most often misunderstood. So communication is a big part of that, and simplification of message is a big part of everything we do. I'll just leave it there.
Great point, thank you. So let's go to the audience. I think Julia and then Sabrina. So Julia, please.
Okay, so my question is kind of in line with what you were talking about right now, kind of about the dialogue between science, research, data, and then the the policy part. Because as maybe Carolyn, and I think before it's been mentioned as well, for example, with Quebec's decision to space out the doses and all this, I feel like we've kind of seen a tug of war, or maybe an attempt at balance between trying to follow what's proven and data and science being informative, while also, governments trying to do something that logistically makes more sense with the resources that they have, while also being in kind of uncharted territory. So I just was kind of curious about how maybe your opinions on that balance and the push and pull between these two sides.
I can kind of continue where I left off. It's the same answer, is that understanding all of the different stakeholder mandates and fears, and understanding and motivations are critical. And the more we touch on topics that affect greater population, I don't think there's going to be another one in our lifetime at this scale, but the more we touch on things that affect more people, the more stakeholders involved in the decisions are going to be around the table. And so I come back to the same thing. Carolyn made a great point about collaboration. For us in developing medical tech involves significant collaboration with different people who speak different language. It always has, but now that dynamic is forefront in this discussion. So collaboration and communication we can't get anywhere without a very specific type, because not everybody speaks the same language. And you talked about policy, Julia, but policymakers don't have all of the information they need, or the knowledge combined with the information to be able to make the right decision. And what is the right decision? Is public health, mental health is not being talked about. The economy is not being talked about. 3 or 4 months into the pandemic, it was one thing to focus 100% on public health, but 15 months in, I think these other topics are very important too. How do you balance that? There's no shortage of challenges here, but I think it comes down to access to information, open-minded collaboration, and communication again. So the soft soft side of the science.
Right. Yeah, maybe so I'll just have a few observations on that too. And for me, the Quebec response has been almost a split personality. So on one hand, and I think with the vaccine, that's exactly the right response they had, right? So you make a risk-benefit analysis and you decide what is, and then you go, and you can still adjust later on. And I think there's this famous video on YouTube also about one of the WHO experts who tackled Ebola and all this disease. When you have an infectious disease, you got to keep moving, right? You got to keep, if you just wait for all the data to be there, if you wait for the enemy to be basically you're knocking at your door, you're toast. And that's kind of what we're seeing in some ways. And so I think for vaccines, they did the right thing. And I think it seems like it's paying off. And then on the other hand, I said split personality because for rapid tests was exactly the opposite. It said, oh, we don't know. Oh, it could be that people will have bad reactions, or people might not do. And it's like, they invented all these obstacles instead of trying to roll them out and use them, because they can be, there's data, and now there's also really on the ground data showing that they can be extraordinarily powerful. So it's why in one case this way, why in the other case, it's kind of also in an interesting sociological study field here.
Okay, I think Sabrina had her hand raised. She seems to have disappeared. Sabrina, if you're there, feel free to turn on your microphone.
Oh, it was just similar to the last question, so I'll let someone else ask. Thank you.
Sounds good. Okay, Halima, please.
Yeah, I just had a question about like the messaging around vaccines, in particular the safety. So there has been some mixed messaging around particularly like the AstraZeneca vaccine. And I've seen people on Twitter talk about like buyer's remorse after like a first dose, for instance. So how would you suggest that the scientific community combat like the mixed messaging and now like the infodemic, quote unquote, that we're facing around the vaccines and other aspects of the virus?
Such a great question. And I don't think there's a very easy answer, but I can tell you that, and I saw Carolyn and David both turn off your mic, so I'll pass it over to you right away. But I can tell you that there's a lot of fear, and there is a ton of media, and there's a ton of news that we never had before. I think if you had the clinical trial reports of almost any pharmaceutical, you would never go into the pharmacy again. So, the people that are in charge of making these decisions manage risk benefit. And that's what our entire system is based on. And if you focus only on one aspect of that, which is a very convoluted and complex equation, well, you can, spread a lot of fear or make the wrong decisions or sow confusion. I'll leave it at that.
I think it's a really very important point, and it comes back to this balance on how much, and we've seen it, I think, with masks, where people at some point say we shouldn't have masks because they didn't want the public to take masks, and also like with this imperfect information and giving the information to the public without actually sounding too convinced and too ideological about it. And at the same time, you have this misinformation campaigns. That's certain. I see that with my parents, which are pretty good, and they got the vaccines, but then they're reading, it could change you or whatever. They were starting to question that. Or actually, even here locally, we had now the NACI, the National Panel on Vaccine, who basically kind of made this claim that, or made this statement that the RNA vectors are better than the AstraZeneca vaccine. They're preferred, right? Well, I got an AstraZeneca vaccine, so what, did I get a second-class vaccine? And actually, there's no significant, because at the same time, there's actually studies in the UK that show that the efficacy of these vaccines is totally indistinguishable, both in terms of getting the infection, of transmitting the infection. And so it was maybe a lot of, because like the earlier, the RNA vaccines, they were tested early on before the variants were there, whereas the AstraZeneca vaccine was tested in the UK when there were already lots of variants circulating, right? And so this communication aspect is, and it's small things can then get amplified, and then of course the opponents will seize on whatever question they have to amplify. And so I think we have a role in terms of us as scientists or biomedical engineer, a role to play, and we have also the capacity and the ability to dive a bit into these questions, inform ourselves, and help people address some of the people's questions here. That's what I would have to say. I don't know, Caroline.
Yeah, I guess from my perspective, this comes back a bit to one of the earlier questions about, as scientists, we're not necessarily always trained in communicating our findings. And I think that I hope that this is a lasting impact of the pandemic, is you have to learn to communicate what you've found. And I think like, for instance, about the vaccines, I think first of all, you have to recognize that people's hesitancy or concerns, this is not like quote unquote uneducated people or like, you know, specific subsets of the population. This is across the board. There's tons of people with fantastic educations who know a lot about the subject who express hesitancy. So finding a way to reach everyone in a way that's informative and sort of respects people's hesitation is going to be like key. And also just communicating some realities. Like, for instance, there's Amin Kamen who in our department who works in vaccine development, and I'd never heard this before, but he said, look, usually you develop a vaccine and the cases of that disease are so low that it takes a long time to just generate enough data on whether or not you had protection. And now you're literally giving out a vaccine while this is like rampant. People are getting infected all the time. It really just narrows that time window for how long it takes to hit that target of how many people were infected versus not infected in the two classes. And hearing it said that way, I think it changes the story a lot of the question of why were they developed so fast? Is there something that was sort of done in a shady way? That was sort of a really great explanation to me, something that made a lot of sense. And so having people with that knowledge who can communicate like that, I think is going to be really important. But then the last thing I would say is, and I think it was already touched on, science is not, you know, science changes. What people were saying at the beginning is not that the science is changing, but our understanding of the science is changing. And so being at least receptive and open to that and communicating what you know at the time, I think is very important too.
I'll just add something quickly, Carolyn. I think you made a great point. This is the first time in our lifetime, you know, that every scientist and every regulator in the world is looking at the same problem. So naturally, things are going to happen very differently than they would in a different, you know, less intense scenario. And the other thing that I thought was encouraging about the AstraZeneca story was that there were some blood clots, there was a response, there was a a reaction in the market, and then there was some we studied data and looking at it, and I heard one of the quotes from an AstraZeneca rep saying that shows that the process is working. You know, in regulatory we call it post-market surveillance, and when the general public sees that, it's very frightening. When we see that, we think the process works. And again, to Carolyn's point, we're seeing it accelerated, you know, 100-fold. But that should, again, miscommunication between the science world and the general public, that should raise confidence in the entire process. Unfortunately, it brings a lot of fear, which you can also understand, right? And I'm sorry to cut out. I do have to go. I just want to thank you again, Jesse and David and Sabrina. Thank you for inviting me, and take care, everybody. Thank you.
Thank you for coming.
I have to get going too. Sorry.
Okay.
But thank you very
You're all
much.
leaving. So I'll do the closing word then.
Okay. Bye, everyone.
Bye. Thank you.
I'll have to run to, yeah. So just to come back, and I think hesitancy is actually legitimate. It's this pandemic. There's so much information. These are, especially the viral RNA vector vaccines, are experimental vaccines. They have never been tested. So it's fair to ask, is this really going to be safe? And I think, we don't really know the long-term outcomes, but we know enough about vaccine and the fact that you actually exposed every day that, the chance that this would have an effect on you is very, very small. And I think someone put it in some place like, if all vaccines were given in a red room, and now you give the vaccine in a blue room, well, someone could say, oh, you don't have data on that. Well, we don't have data, but it's not really relevant data, right? And so in some ways that applies also to some of these vaccines. We don't know these experimental vaccines, but we know generally about vaccine a lot. We know about the immune system, and this is relatively consistent in how it develops. So this could then basically allay some of the fears that people might have. Okay, but I think I'll stop it here too then, and I'll give it back to Jesse. I think.
Okay. Unless there are more questions from the audience, I think it's about time to wrap it up. I want to thank you, David. You're your last one here, last man standing. So thank you. Obviously, we thank all our panelists, and thanks to everyone in the audience. This has been very insightful for me. I hope for everyone else. And have a nice day.
Awesome. Thank you so much, Jesse, and thank you so much to all for panelists. Unfortunately, most of them left already. But yeah, thank you very much. It was a great panel discussion. So I guess now we can go with our next part of the program. And this is poster presentations. So if you haven't had the chance to visit posters, you can do it. You can do it right now. And we're going to be opening breakout rooms. So poster presenters can join their own breakout room and we can just chat around. We're going to be here for like 40 minutes. So we can just use it as a networking time, visit posters, just discuss with your colleagues. And also don't forget to vote for your favorite poster. The deadline is at 4:30 p.m. today.
Can I ask how we can vote? Because I didn't find it.
Oh, I just sent the link in the chat, but I'll also send it in the FeedLoop chat.
Okay, that's good.
Okay, just 1 second. Oops. And if somebody from the council can share their screens with the instructions, that'd be great.
I will do that. Can you make me co-host, please?
Oh, yes. I don't know why it overrides privileges.